begin. Sorry about that. All right, so we are all familiar with COVID-19 infection caused by SARS-CoV-2. We are all familiar with the catastrophic event that followed with this pandemic. But today I'm going to talk about a novel aspect of this infection. Does this infection increases your risk of cancer? Could this be a problem in future? Because we know that at least 7 different kind of viruses have been associated with various kind of cancers, and there are so many intersection in the pathway of pathogenesis of this virus and cancer signaling pathway that raises this concern, and that's why I'm interested to talk about this today. So a brief introduction into the topic. COVID-19, as we know, is caused due to SARS-CoV-2 infection, which was declared a pandemic by WHO on March 11, 2020. As of December 21, 5.94 million deaths were reported, which is just an estimate, and actual number could be higher. And this is an actual old data, but just to emphasize the impact of this disease on the entire human population. According to WHO bulletin, infection fatality rate was 1.63%, which is not that high, but still the total death toll was astounding due to pandemic nature of this disease. COVID-19 infection can be asymptomatic or can cause multi-system disease. Most frequent symptoms include like fever, cough, headache, sore throat, diarrhea, tiredness, loss of smell, taste. Severe symptoms include ARDS, which is like acute respiratory distress syndrome, which is very severe form of this disease caused by severe inflammation of the lungs triggered by cytokine storm. Stroke, cerebral venous thrombosis, myocarditis, stress cardiomyopathy, arrhythmias, they all have been reported as cardiovascular and cerebrovascular complications. Now, a subset of patients continue to experience different kind of symptoms beyond 4 weeks from actual infection, and this was termed as long COVID or post-COVID syndrome. Now, we don't know the exact pathogenesis at this point, this phenomena is really interesting and important because there is quite a bit of similarity in the pathway of this pathogenesis of this syndrome, post-COVID syndrome, and carcinogenesis as well. That's why this is relevant in this context. Suggested hypotheses include persisting reservoir of SARS-CoV-2 in the tissues, immune dysregulation, possible reactivation of viral pathogens such as Epstein-Barr virus and HHV6, priming of immune system for molecular mimicry. In a recent article published, I think a few months ago, a group of researchers found that there was like some autoantibodies against autoantibodies of ACE2 inhibitors circulating in our body, which again is like something on the line of molecular mimicry and immune dysregulation. Now, why do we think this carries an oncogenic risk? So the thought here is there is an intersection in the cancer signaling pathway and the inflammatory pathway, and based on the existing knowledge of interaction of SARS-CoV-2 with host at cellular level, there have been several interactions noted between inflammatory pathway and the cancer signaling pathway, which obviously raises concern. 15.4% of all cancers can be attributed to carcinogenic infections for which viruses are the main risk factor. So on that note, do we think SARS-CoV-2 can carry a similar risk? SARS-CoV-2 can potentially increase the risk of lung, colorectal, breast, pancreatic, and oral cancer, according to some studies, and I've included a reference for that. So a quick, talk about the pathogenesis to understand better the process here. So SARS-CoV-2 is a positive sense single-stranded RNA with spherical capsids. Cellular invasion happens through interaction of spike protein on the viral membrane, which binds with the receptor domain of S1, receptor binding domain of the S1 subunit with ACE2 on the host cell surface. After cellular invasion, replication and transcription of viral genome occurs in cytoplasm of host cell. Now, is it possible that the reverse transcribed RNA genome of SARS-CoV-2 can integrate with human genome? That is very highly controversial at times because there are so many conflicting evidence, and this missing piece of information may be highly relevant in future, as certain oncogenic viruses such as Hep B and hepatitis HPV are known to cause oncogenesis through activation or inactivation of certain host genes, oncoproteins, and tumor suppressors. Hep B, in particularly, its DNA integrates directly and can regulate crucial intercellular pathways such as cell cycle regulation, preventing apoptosis, cell-to-cell interactions, cell signaling pathway, and all these can mediate the acquisition of pro-oncogenic properties. Now, if we talk about what could possibly be the underlying mechanisms for oncogenesis because of SARS-CoV-2, then there are a few theories. I'm going to go over the most commonly talked about one at this point, or whatever piece of information we have. So the first one is viral mutagenicity. So viruses, we know, can regulate cell cycle, damage host DNA, use host translation machinery, disrupt apoptosis, suppress immune response to evade immune detection and destruction of mutated cells. Now, viral infection can activate proto-oncogenes or disrupt tumor suppressor mechanism. SARS-CoV-2 actually can inhibit tumor suppressor gene P53 and PRB and their proteins. PRB is the gene that we commonly talk about when we talk about a double hit theory. NSP15 is an endoribonuclease encoded by all the coronaviruses, so that includes SARS-CoV-2. It can affect cell cycle by increasing the proportion of cells arrested in the S phase of cell cycle. How it works is SARS-CoV-2 may induce expression of NSP15, which acts on tumor suppressor PRB through LC-XCE motif. This interaction between NSP15 and PRB causes proteasomal digestion of PRB. In addition, it can suppress P53 as well, and through its NSP3 protein, which results in stabilization of RCHY1 ubiquitin E3 ligase that targets actually P53 and causes degradation. Now, this leads to a lower level of P53 in the cytoplasm, which is really a predisposing condition for developing cancer. Now, SARS-CoV-2 can inhibit DNA damage repair. N protein interacts with LARP1, which is dysregulated in various cancers, especially those related to viral infections, and appears to cause tumor. Well, this is another clue towards how SARS-CoV-2 can affect and increase your oncogenic risk. SARS-CoV-2 interacts with various epigenetic pathways similar to oncogenic pathways such as NSD2 and HDAC2. NSP8 actually interacts with NSD2, and HDAC2 interacts with NSP5. So what I'm trying to say is there is a lot of interaction of proteins encoded by SARS-CoV-2 with the oncogenic pathways, which has already been studied in other kind of cancers. Similarly, protein E is a envelope protein associated with BRD2 and 4, which is involved in carcinogenesis by activating proto-oncogenes such as MYC. Inflammatory cascade. So as we know, COVID-19 can induce very strong inflammation in the host through multiple mechanisms. Interleukin-6 has been so far the most extensively studied cytokine, which has been associated with chronic inflammation in call it autoimmune disease or cancers. We have extensively tried IL-6 inhibitor in treatment of COVID, tocilizumab, because to suppress inflammation. IL-6 plays a key role in hyperactivation of NFK beta and activation of STAT3. STAT3 activation can cause G1 to S cell cycle transition along with induction of proto-oncogenes C, MYC, PIM1, and PIM2. Now, TNF alpha can trigger NFK beta activation, which causes DNA damage, chromosomal instability, mutation through production of reactive oxygen species, induction of activation, induced cytidine, deaminase, prevention of apoptosis, which can all lead to tumorigenesis. And we already know that Epstein-Barr virus exerts their oncogenic effect by stimulating NFK beta pathways. So that's another evidence here. Interleukin-1 beta, TNF alpha play a key role in upregulation of heat shock protein 27, which has been observed to be upregulated in various cancers. And HSP27 can inhibit multiple steps involved in apoptosis. Immune dysregulation. COVID-19 infection triggers innate immune response, leading macrophage and other immune cells to generate large number of cytokines such as IL-6 and activates JAK-STAT3 signaling. Now, IL-6 JAK-STAT3 signaling is abnormally activated again in various types of cancer and plays an important role in suppressing the anti-tumor immune response. This has been actually extensively studied in lung, liver, and breast cancer. In fact, there are some novel inhibitors of IL-6 JAK-STAT pathway, which including the STAT3 selective inhibitors, which are currently being developed. So STAT3 hyperactivation in tumor cells can occur as a result of increased IL-6 level, which promote angiogenesis, metastasis, and down modulation of anti-tumor immunity response. STAT3 has been studied and has been found to have a role, identified in breast cancer metastasis. Now, some more talk about the other, pro-inflammatory proteins. So type 1 interferon are involved in development of innate and adaptive immune response against both cancer and infectious disease. However, type 1 signaling was dampened in patients with severe COVID, and a low level of IF interferons along with an increased level of IL-6 was noted again. So it's like, you are losing protection and increasing risk here at the same time. So obviously that predisposes you to oncogenesis. Dysregulation of angiotensin 2 AT1R pathway contributes to production of inflammatory growth factor, cytokines including 1-alpha, 6, 8, and MCP1, and macrophage colony stimulating factor by inflammatory cells. Generation of reactive oxygen species by neutrophils, macrophages after the angiotensin 2 AT1R activation in the inflammatory microenvironment causes extra damage by its deleterious effect on the DNA structure. So some evidence here regarding damage to the DNA itself, and obviously we know, like I mentioned earlier, SARS-CoV can prevent DNA damage repair as well. Now, persistent infection and antigenicity. This is a very interesting concept here, because the pathogenesis of long COVID has certain resemblance here in the same way you have a persistent viral infection, like certain persistent viral infections such as Hep B and Hep C obviously has been established to cause hepatocellular carcinoma. So the exact pathogenesis of long COVID is not clearly understood at this point, but there are various hypotheses, including persistent reservoir of SARS-CoV-2 in tissues, immune dysregulation, reactivation of underlying pathogen. So SARS-CoV-2 nucleocapsid proteins has been detected in multiple extrapulmonary tissues such as liver, gut, lungs as well. Persistent viral antigen or RNA in gastrointestinal tissue and fecal excretion has been noted as well. So now the persistent infection is sort of unlikely, but persistent antigen expression is what is suspected, and that is something which can trigger chronic inflammation, which can again lead to higher cancer risk. SARS-CoV-2 spike and tumor suppression protein may have autoimmune cross reactivity, which could be a potential mechanism underlying prospective cancer insurgence. Concept of high molecular mimicry has also been reported between spike glycoprotein. So let's talk quickly about some individual cancers here and how SARS-CoV-2 can play a role there. So cytokine storm triggered by SARS-CoV-2 leads to pulmonary interstitial fibrosis, and it is a chronic inflammatory state, which is a known risk factor for lung cancer. Continuous damage to alveolar epithelium leads to hyperplasia, metaplasia, fibrosis, which stimulates incidence of lung cancer. ACE2 modulates angiotensin 2 expression, causing heightened expression of NFK beta regulated inflammatory cytokine, activation of P13K AKT pathway, known factor for tumor development. This virus can also cause suppression of immune system by NK natural killer cells and cytotoxic T lymphocyte cells exhaustion, and thereby evading immune response. Interleukin-1 beta 17 have carcinogenic effect by stimulating epithelial cell proliferation and tumor growth. Again, TGF-beta, interleukin-6, galectin-3 triggers transformation of myofibroblast from endothelial cells. Similar mechanism has been noted in tumor microenvironment of non-small cell cancer. Pro-inflammatory cytokine triggers oncogenesis, oncogenesis through mechanism including DNA damage, increased amount of angiogenic pathways such as VEGF, IL-8, ICAM-1, VCAM-1, induction of cell proliferation and prevention of apoptosis. Similar risk has been also studied in colon cancer. Studies show that it reduces, it causes a gut dysbiosis, and then it reduces the number of butyrate protease of short chain fatty acid producing bacteria. Reduced butyrate producing bacteria leads to decreased level of IL-22, which is really important for integration of gut and lung epithelial barriers to prevent antigenic priming and immune mimicry. Similarly, it has also been been studied and being actually studied in context of other cancers such as pancreatic adenocarcinoma, increased EMT in breast epithelial cells due to downregulation of E-cadherin expression and upregulation of N-cadherin and the SNAIL1 by gamma variant. This has been studied in the context of breast cancer. So I understand, like at this point, every information, everything is pretty much hypothetical, but that's where we begin. So what's the future direction, and what do we need to do in future? So obviously we need more, studies into the cellular and tumor microenvironment, pathogens of SARS-CoV-2 to get more information. These are right now all we have is like bits and pieces of information. So we need like more data to put it together to see the clear picture. We definitely need to do like cross-sectional studies in future, like 5-10 years from now, to see and analyze if there's any increased to explore any association between cancer and COVID-19. And should that happen, then do we need new screening guidelines? So these are all some questions to be addressed in future with more research. These are some references I have added. And thank you for listening. I'll take your questions now.